Okay. So I guess it's my great pleasure, it's my great pleasure to introduce Jean-Michel Boudreau, who's going to give a talk on his master's thesis work. And yeah, we're looking for some interesting new insights in how bacteriorhodopsin behaves under light. Jean-Michel, over to you.
Hey, everybody. Yeah, so I'm Jean-Michel Boudreau, as Dwayne just introduced me. And some of you may know me, judging from my very outdated LinkedIn's increase in views. So I'm a master's student in Dwayne Miller's groups, and today I will be presenting on the AC Stark effect on the electronic structure and single photon absorption of bacterial rhodopsin. So before we get into any of the details, I'd like to lay out the organization of my presentation that you'll be seeing today. First, I'll be talking about some of the progress that others have made in understanding the phototrigger dynamics of bacteriorhodopsin. Following this, I'll address one of the ongoing issues in preparation of an initial state of bacteriorhodopsin. I'll frame this problem under the context of this protein. However, it can apply in the study of any molecule that requires laser excitation prior to studying properties of interest and its dynamics. After this introduction into the research at hand and the objective in the research I did, I'll present briefly on how the Stark effect can be calculated to a first order approximation using time-independent perturbation theory. I then give a brief discussion on general Born-Oppenheimer molecular dynamics. I define it here as a framework for mixed quantum classical dynamics, in which the electronic structure and classically defined nuclei are evolved in an alternative fashion to capture the total dynamics of a system. Together, the equations I show should provide enough theory to understand how the Stark effect was calculated in a quasi-time-dependent manner. Furthermore, I will present how the model and the program I used came to fruition. Lastly, I go into the results of the calculation I did, and in particular, I will show how the effect of the time-varying electric field upon the geometry of the molecule, and also the linear Stark effect on the electronic structure and transitions of the bacterial rhodopsin model system. So without delay, let's get into the context of this work. So the rhodopsins are a class of membrane proteins that harvest light to generate a plethora of functionalities, including vision signaling, ion channeling, and also in actuating neural cells for optogenetics research. Each member within this family is conferred its light-triggered functionality via a retinal chromophore that undergoes rotation about a single pi bond along its conjugated alkene tail upon photoexcitation. The quantum yields, regiospecificity, and the timescale for this photoisomerization, however, differ depending on the environment of the retinal chromophore. In particular, the set of steric constraints, Coulombic interactions, and the electronic structure changes induced by the respective protein and signature environments dictate the dynamics of this process. This ultimately manifests as the versatile set of functions I just mentioned on the previous slide. And arguably the best characterized member of the rhodopsin family is bacteriorhodopsin. This protein is found in haloarchaea and is used to generate a proton gradient by utilizing light energy to pump protons out of the cell. It does this to drive an ATP synthesis by an ATP synthase protein located on the cellular membrane. So the photoinduced dynamics of bacteriorhodopsin have been characterized in detail by various time-resolved optical methods. When combined with site-directed mutagenesis and theoretical methods, these optical approaches have proven very powerful in converging upon a widely accepted sequence of bacteriorhodopsin spectral intermediates. Upon absorbing a single photon of approximate wavelength of 535 nanometers, the retinal moiety in the all-trans conformation undergoes an electronic structure change such that the electron density is donated from the beta ionone ring terminus of the alkene chain right here to the shift base region over here. So subsequent depopulation of the Frank-Condon region is initially driven by a stretching mode, which inverts the sigma pi bond order occurrence along the alkene chain. So for instance, these single bonds here would show more double bond character, and the double bonds here would show more single bond character. And this leads to an intermediate right here known as the I-460 intermediate. Following this, there is a concerted twisted deformation of the three bonds between the C11, C12, C13, C14, and the C15 and nitrogen bonds here. And this directs the nuclear configuration through a conical intersection towards the vibrationally hot J625 intermediate here. And continued motion ultimately leads to the 13 cis photo product, typically denoted as the K590 intermediate. These changes in the retinal environment as a direct cause of this isomerization process then sets off a cascade of conformational changes within the protein to facilitate the transport of a proton from the retinal shift base to the extracellular side of the membrane. The retinal then undergoes reprotonation and thermally re-isomerizes back to the bacteriorhodopsin ground state. After which, another photon absorption event may occur to kick off this photocycle again. Though considerable effort has been made into elucidating the various aspects of the phototrigger dynamics of bacterial rhodopsin, there remains a great deal of dissent with respect to whether the experiments concerned with the dynamics accurately reflect the in vivo light harvesting process. In particular, the high powers used to pump the system and obtain appreciable signal put the validity of the interpretation of the data into question, as nonlinear optic effects such as multiphoton absorption and the Stark effect will influence the behavior of the system. Unless accounted for, the interpretation of the data may have some errors. So for example, consider the following study published by a team of scientists at ETH in Zurich in Science 2 years ago. So Nogli et al. Published an article on the retinal isomerization process in bacterial rhodopsin using an X file to capture snapshots of the molecular dynamics on a femtosecond timescale. The laser power densities used, however, were at magnitude upwards of 324 gigawatts per centimeter squared. This magnitude is tenfold greater than the current estimates for the onset of Stark effects to appear in the electronic absorption spectra. Now, just to provide some context to these numbers, the global air mass 1.5 reference solar spectrum has an irradiance of about 100 milliwatts per centimeter squared. Now, some of you may be wondering what exactly is the Stark effect, and I know from my personal experience, this was a completely unknown term to me prior to the start of the last school year when I started this project, so I'm just going to provide a slightly from description. Well, I'll do that later on, but just to make sure we're on the same page, I have a qualitative description right now. So it is essentially a shifting and splitting of spectral lines due to the coupling of an external electric field with the molecular system of interest. Here, I'm just showing the shifting of electronic absorption lines. So as all of us know, spectral lines and electronic transitions match the differences in energy between two different electronic states. And when an external electric field is present, which could be from the environment of the molecule we are studying, the electronic states will change in energy, which will cause the electronic absorption spectra bands to shift. Furthermore, the orbitals of the electrons will be deformed such that they are now biased towards the positive terminus of the electric field. So in other words, the electron density will be shifted towards the positive end, as you can see here. Prior studies have predicted the Stark effect on the vibrational spectra of retinal at the Austin Model 1 level of theory. So using a static external electric field, strength of about 1 terawatts per centimeter squared, directed near parallel to the dipole moment of retinal, Marty et al. Simulated a simplified yet good simulation of the environmental effects in proteins and found a large increase in the simulated intensity of the CO stretching and the C11, C12 stretching vibrational modes in red now when compared to the field-free simulation. So the effect is particularly strong in the CO stretching mode, increasing by more than an order of magnitude, as can be seen in the necessity to change the order of magnitude in the y-axis. To further understand the impacts of the linear Stark effect on the properties of bacteriorhodopsin, we performed a suite of calculations on a model bacteriorhodopsin system using an in-house developed program. So in particular, the work you are about to be presented with addresses the linear Stark effect arising from an ultra-fast pulse laser. The program consisted of a set of Bash and Python scripts that can be run either on a local host or on a Slurm workload manager as used by the Compute Canada clusters. It works by interfacing with a copy of Games US to solve the electronic structure and finding the gradients with respect to the nuclei in the presence of an external electric field. These simulations you're about to see are performed exclusively on the ground state. The nuclei at each time step is propagated by application of those gradients within an ODE integration scheme, and for these calculations, I used Beeman's algorithm. The technique of solving the electronic structure and gradients, followed by updating the nuclear positions reiteratively, is sometimes denoted as the Born-Oppenheimer molecular dynamics framework. The electric field of the ultrafast laser pulse is modeled in our calculations by an approximately 1 terawatts per centimeter squared Gaussian envelope pulse with a wavelength of approximately 535 nanometers. Three respective trajectories corresponding to the Cartesian axis in which the laser pulse oscillates in were simulated for 300 femtoseconds. Density functional theory was used to solve the electronic structure throughout the molecular dynamics simulations, and furthermore, linear response time-dependent density functional theory was used to characterize the visible spectra of bacteriorhodopsin at each time point along the simulation. Electron hole analysis was carried out using multi-WFN 3.7 at certain time points during the simulation in order to determine the evolution of the electron and hole character of the moieties in the model used. I also used VMD on the multi-WFN outputs to visualize the molecular orbitals as well as the distribution of the electron and hole. Lastly, I also developed a few Bash and Python scripts to visualize some of these properties over time. Now, to jump brief into the theory, I want to go into how the Stark effect was calculated, in particular the linear Stark effect. So you may have caught a glimpse of it a few slides ago, but I'd just like to touch upon it a little more. And for all the theory involved, it is assumed we will be using the Born-Oppenheimer approximation. So suppose a system of interest is perturbed by some external electric field we will denote by the Greek capital letter Z. Approximating the total energy of the system by Taylor series expansion yields equation 1 here. If we take a look, we may see a term we recognize, namely the dipole moments. It is simply the derivative of the energy with respect to the electric field evaluated at zero field strength. The expectation value of the dipole moment is simply the right-hand side of equation 2 with the dipole moment operator sandwiched between the rho and ket for the electronic state. Now, if we truncate the Taylor series expansion of equation 1 after the first term, the total energy of the system perturbed by the external electric field to a first order approximation is given by equation 3 here. Recasting the equation in terms of a perturbed Hamiltonian yields equation 4. Note, though the form as is of a static dipole electric field, a quasi-time dependent electric field may be generated by means of applying a time-varying electric field within the Born-Oppenheimer molecular dynamics framework, particularly when determining the electronic structure and the gradients with respect to the nuclear coordinates. Okay. Now, to get a quick intuition for this type of molecular dynamics, I will simply present the algorithm for running Born-Oppenheimer molecular dynamics and treat each step as a black box. So first, the electronic structure of the system's initial starting point for dynamics must be calculated. Following this, the gradient with respect to the nuclei is then calculated, and we use Games US to accomplish these two steps. Then the nuclear coordinates are updated by application of these nuclear gradients within Bieman's algorithm. The electronic structure and gradient is then recalculated using these new coordinates. Next, the velocities are updated with Bieman's algorithm. After that, we repeat steps 3 to 6 in a loop until we reach our desired endpoint. And so basically for every loop, we're applying a different external electric field strength with its magnitude at that particular time point dictated by the equation for a Gaussian envelope time varying electric field. And we do this to represent an ultra-fast laser pulse. In the calculation used to obtain the results I will present, the time step starts off at 1 femtosecond. However, due to the increase in change of magnitude of the electric field over time, the time step is lowered to 0.6 and 0.4 femtoseconds during some intervals. And this is just to increase the resolution of the properties of interest. So now that that's over with, we have a pretty good understanding of how the framework for the molecular dynamics one was used to capture the linear Stark effect. I still haven't provided any context as to the model. So to create the model, I extracted the bacterial rhodopsin coordinates from the RCSB PDB databank and created a model similar to that by Kovacs et al. In their 2019 Nature paper. UCSF Chimera was used to add hydrogens and truncate the number of moieties I included in the model. So the amino acids and the water molecules chosen for the model were all in the vicinity of the retinal in the active site of bacteriorhodopsin. The amino acid members consisted of tyrosine 57, arginine 82, aspartate 85, tryptophan 86, threonine 89, tryptophan 182, tyrosine 185, aspartate 212, and lysine 216. All amino acids were cut at their alpha carbon positions inclusive, except if that alpha carbon lay between two adjacent amino acids. And furthermore, the water molecules included in the model were of water 402, water 405, water 407, and 418, as these were part of a known hydrogen bonding network within the active site. 3 of the waters were included in a known pentagonal hydrogen bonding network, as you can see here. And of course, we also included the chromophore retinal inside the model. Avogadro was then used to adjust the relative positions of the hydrogens of the water molecules such that they appeared in good locations for hydrogen bonding. And this final model consisted of 208 atoms with a 0 net charge. The geometry was optimized at the B3 LEAP D3 DEF2 SVP level of theory using the cluster approach in Games US. And the cluster approach is basically, I locked all alpha carbon positions in place during the entirety of the optimization procedure, as well as the dynamics, to prevent artificial movements of the placement of all moieties. And furthermore, I use the conductor-like polarizable continuum model, here the CPCM, as you can see on the fourth step there. I use the polarizable continuum model to simulate an implicit protein environment. The de facto standard relative permittivity for treatment of a protein environment is 4, so that's what I did. I chose to use the B3 LEAP functional and DEF2 SVP basis set as a recent benchmark of vertical excitation energies of biological chromophores published this year gave pretty solid results. And so I use this combination for the geometry optimization part of this study and other parts of this study as well. The final geometry optimized structure are the ones you are viewing in this slide. So like I said earlier, we developed an in-house set of scripts to interface with Games US to carry out the trajectories. None of the 3 programs I found were able to do the Born-Oppenheimer molecular dynamics with a Gaussian envelope time-varying electric field, while also locking the coordinates in place. So I chose to create one that can do it instead. The slide you're seeing represents the flow for the scripts I created using Bash and Python. I didn't have a personal computer with the resources to accomplish what I set out to do with bacteriorhodopsin, so I mainly tested out on a water molecule and then adapted the code to be run on Compute Canada clusters, which require the Slurm workload manager to submit the jobs. I chose to begin with a bash script because it allows the easiest manipulation of the environment and text parsing, in my opinion, for a Linux environment at least. So first, the program requires the user input for running the molecular dynamics in presence of a time-varying electric field. So these parameters include the time step, the end time, and both of those are in femtoseconds. It needs a molecular dynamics algorithm to be used, which is the Beeman's algorithm. It also can be used with velocity roulette. The parameters for a Gaussian envelope laser pulse are also needed, and the strings specifying the atom numbers to keep locked in place, if you would like to keep some locked in place. So the parameters for the Gaussian envelope laser pulse are the max electric field strength in volts per nanometer, the frequency of the pulse in in petahertz, and the full width half max of the pulse in femtoseconds. So the bash script then automatically sets all necessary environmental variables and creates all directories for running the molecular dynamics using the relative path of the main directory you have. The driver requires a co-located games US input file to start the dynamics or restart the dynamics from the last time point. So it creates some parameter files by reading the initial games US input file you have and uses them to create the next games US input file for future iterations of the dynamic simulation. The string type arguments that you pass during this step in the initial parameters for the dynamic step is passed to the a Python script, and the Python script recasts these strings as other type of variables for calculation. It also imports all environmental variables created in bash and begins a molecular dynamic simulation. The numerical integrations are handled by NumPy library, and I initially also had NumPy to increase the speed of the calculations, but I had issues running on Compute Canada, so that was just removed. The Born-Oppenheimer molecular dynamics portion was written with the algorithmic steps I stated prior. So first, the coordinates are updated, and also we calculate the electric field strength, and both these parameters are placed into a new games US gradient job, which calculates both the electronic structure and the gradients with respect to the nuclear coordinates. The velocities are then updated, and all the coordinates, velocities, and gradients are saved to respective directories if you want to use them for analysis, or also if you need to restart the calculation from the last successful time point. And then this simulation continues until the final time point it reached, and when it realizes that, then the simulation is over. So each of the, to reiterate myself earlier, 3 simulations using these scripts were carried out, and each of the 3 respectively correspond with the Cartesian axis in which the electric field oscillated in. The dynamics were carried out at the same level of theory as the geometry optimization. So again, this is a CPCM, B3 LEAP, D3, DEF2, SVP level of theory. So without further ado, I'd like to get into the results of carrying this simulation out. So here we can see the geometries of the bacteriodopsin model over the course of the simulation. On the top, we just see the directions of the applied external electric field relative to the retinal moiety. And on the bottom here, we can see the geometry at certain time steps. The blue corresponds with the original geometry optimized structure, so at 0 femtoseconds. The red corresponds with the structure at 150 femtoseconds, and the yellow corresponds with that at the end of the simulation. The Cartesian axis direction specifying the electric field direction is given below each of these figures. And as evident from the root mean square of the relative difference of the atomic coordinates over time, the initial structure changes by about 0.10 to 0.15 angstroms in position during the entirety of the simulations. Interestingly enough, the geometry is most susceptible to the time-varying external electric field when applied in the z direction. The electronic structure shows noteworthy differences due to the electric field perturbations with its application in each axis showing unique features. The most significant changes amongst the three separate trajectories are the rearrangement of molecular orbital energy levels due to the electric field. And for instance, the highest occupied molecular orbital is not necessarily fixed, and it alternates between different molecular orbitals during the simulation. Interestingly, too, almost all the orbitals go through drastic changes in ordering and distribution due to the orbital mixing, but the lowest unoccupied molecular orbital, which is this gray line here, it remains relatively unchanged, both in energy and distribution, and I mean relative to the other molecular orbitals. And this is regardless of the direction of the electric field. This molecular orbital is essentially the only virtual molecular orbital associated with the excitation to the canonical S1 state, which in my calculations is predicted as an S0 to S5 transition. The greatest change in energy can be seen when the electric field is applied in the X direction, as this direction is nearly parallel to the dipole of the retinal moiety. Here you see the snapshots of the distribution of this luma over time when the electric field is applied nearly parallel to the retinal dipole vector. So this is in the X direction. We see that this is an antibonding pi molecular orbital and is primarily localized on the retinal. These are, the first 8 electronic transitions of the bacteriodopsin model were calculated at the TdB3 leap def2 SBP level of theory, and the optically allowed electronic transitions, defined here as a transitions whose oscillator strength exceeds 0.1, are the product of electron density primarily being evacuated from MO352, 353, 355, and 356. And this electron density from these orbitals is transferred to the antibonding pi lumo on retinal that I just showed you. Other orbitals involved contribute less than 1% to this excitation, and so clearly these are the main players in this excitation. Clearly these molecular orbitals are localized on retinal, tyrosine 185, and tryptophan 86. So in knowledge of this, I created a smaller model to perform the TDDFT calculations to calculate the electronic transitions so that the computations were computationally tractable. So I included all members of the aromatic triad as well as retinal. So the aromatic triad consists of tryptophan 86, tryptophan 182, and tyrosine 185. And so how I crafted this was done by utility of a bash script to remove all atoms that did not belong to the retinal or the aromatic triad at each time point of the dynamics run earlier. All sp3 hybridized carbon atoms that had missing hydrogen atoms due to my Avogadro, and then I calculated the electronic transition at the TdB3 leap def2 SVP level of theory again. I would have included the CPCM framework, but there was some unknown error with Games US, and I couldn't figure that part out. And similarly, Grimm's D3 dispersion was not included because it cannot be described at the TD-DFT level in Games US. So here we can see the electronic transitions of the more minimalistic bacterial rhodopsin model as a function of time during each of the 3 trajectories. The red curve corresponds to the electronic transition with the highest oscillator strength, which is not necessarily the S1 state. As you can see here, it kind of bounces to another state. So the AC Stark effect on the excitation energies corresponding to the S0 to X1 excitation is strongest when the external electric field is applied in the X direction. And this is due to the stronger interaction of the electric field in this direction with the dipole of the retinal moiety. When the electric field is applied in this direction, it's either parallel or anti-parallel with the dipole vector of the retinal and will cause the most significant changes in the magnitude of the energy. As you can see, it's pretty clear. So unsurprisingly, when the electric field is applied in this direction, it also causes the most significant changes in energy of the antibonding pi lumo, as I previously mentioned. Since this first LUMO is a predominantly involved virtual MO in this excitation, the larger changes in its energy would manifest relatively larger changes in the excitation energy of this transition as well. Now, going back to this red curve, one noteworthy feature I'd like to bring to your attention is the propensity of the electronic transition with the highest oscillator strength to change regardless of the direction of the electric field, particularly to those in the same energy regime as the canonical S0 to S1 transition. So to reiterate myself earlier, the transition with the highest oscillator strength is given by this red curve, and we can see that it kind of bounces between different states or different state transitions. And over here, it's really small, but there is it happens at higher electric field strengths. So, for example, looking at the graph in the y direction, as I said, there's some cusps here, here, and it's bouncing between different energy levels or energy transitions, state transitions. Sorry. So going back to my original objective, we can see that even at lower power densities than used in recent literature, the transition with the highest oscillator strength changes between different transitions. Now, the fact that these calculations show a shift in which electronic transition is of highest oscillator strength, and considering that these transitions are similar in energy to those of the canonical S0 to S1 transition, it becomes evident that the linear Stark effect alone necessarily transfers some of the population on the ground state to states other than the biologically relevant S1 state, which is usually the one that's probed. So in other words, the linear Stark effect will change the photochemical pathway being studied from the canonical S1 state to others that may have not been considered. And therefore, potentially erroneous interpretation of results may have propagated in research that did not consider this particular nonlinear optic effect during the initial state preparation if they used high power densities. So now turning our attention to other interesting avenues, here we can see the percent electron hole distribution over time when the time-varying electric field is applied nearly parallel to the dipole vector of the retinal moiety. Again, while the electron character of the retinal remains relatively constant, given by this orange line here, the hole distribution for this transition goes through extreme changes as given by this chaotic nature here. In particular, the moiety dominantly exhibiting hole character for the excitation alternates between all four members included in the more minimalistic bacterial rhodopsin model. Lastly, I'd like to address a threefold near degeneracy, both in energy and oscillator strength in bacterial rhodopsin, when an electric field strength of about 2.49 volts per nanometer is applied away from tryptophan 86. So first, I'd like to show a video of the predicted visible spectra I generated using Games US and the multi-WFN program. So for your general information, the scale here is about 1 second for every 10 femtoseconds of the simulation. We can see the electronic transition energies oscillating back and forth, along with some significant changes in the oscillator strength, with increasingly strong magnitudes of the time-varying Gaussian envelope electric field as time went on. So I don't know how many of you caught that, but at a little over the halfway mark, there is a seemingly threefold near degeneracy in both in oscillator strength and in energy. These three are the ones I'm talking about. Some of the lines are a little faded. So I was curious as to how the electron hole distributions were like for these three transitions. So I ran the Games US calculation through multi-WFN to check it out, particularly through an electron hole distribution analysis. And here's what I got after visualizing them through VMD. So the blue here corresponds with the hole distribution, and the green here corresponds with the electron distribution. We can see that there is significant difference in the hole distribution when the field is applied away from tryptophan 86, with this moiety losing some of its ability to participate in donating electron density to the retinal chromophore. The qualitative argument can be given for this, for its weakened ability to donate electron density. And just put it briefly, the tryptophan 86's molecular orbitals should deform with more electron density distributed along the positive y direction when an electric field is applied away from tryptophan 86, as this corresponds with placing a positively charged plate above this moiety. So with the molecular orbitals spatially deformed in this manner, there would be relatively less overlap between the retinal molecular orbitals and the tryptophan 86 molecular orbitals as there previously was when no electric field was present. The sum of the oscillator strengths of these three near degenerate states, roughly equaling the oscillator strength of the excitation to the canonical S1 state in the absence of an electric field, supports that these threefold near degenerate states are produced from the splitting of the S0 to S1 spectral lines. So now to conclude, I'd like to summarize the main findings of the work that was done. So first and most importantly, the linear Stark effect alone is predicted to cause population transfer from the ground state to states other than the biologically relevant S1 state. And therefore, it requires consideration and interpretation of data when using high power densities to pump the system. Secondly, there's a transition that splits into a threefold near degeneracy, both in oscillator strength and energy when the electric field is applied away from tryptophan 86, and this is of the canonical S0 to S1 transition. Future work should use a more time-dependent formalism to capture the AC Stark effect more accurately, and for example, Floquet theory is one that I came across.
Okay.
Thank you for your time. Before the questions, I'd like to give a formal acknowledgement of my gratitude to my supervisor, Dr. Miller, as well as Olivier Perry-Labrosse and Dr. Semen Maneshi for their counsel in completing this work, and as well as Dr. Mark Wilson for helping me revise my thesis, and also my loved ones. Thank you for your support. And I welcome any questions. Thank you.
Great. Thanks, Jean-Michel. So I guess I open it up for questions.
I have a question. Could you go back to where you first used the start effect, like 9 or something like that?
I'm sorry, it's choppy a little bit.
I think it was slide 9. I'm not sure.
Okay. Yep. Oh, sorry. Is it this one?
Yeah, I think that one. Yeah, yes. So I just want to understand it, complete the start effect. I didn't really get it. So what do we have, like, what is the is that isolated molecule or what? And then you get this split in the isolate. Could you tell me a little more about tell me a little more about this.
Yeah, so this was just more of a qualitative picture of how the linear Stark effect arises. So when it is juxtaposed with this, for example, like when the molecular orbitals are deformed, it changes the state energies, and therefore the electronic transition will also be shifted. These on the side over here are, well, I should have wrote state energies S2, S1, and 0. I'm referring to the energy here. And then the way that it's perturbed depends on the dipole vector of that state, as well when it's dotted with the electric field strength.
So that's within perturbation theory, first order perturbation theory. Or is this exact? I guess this is like, how do we get these?
How do we get these?
Out this state, is that within the perturbation theory? You get them, is that a first order perturbation theory, and you get the energizing state?
Yeah, I'm using these equations here. So when I write, for example, where is it?
Yeah, okay, so you truncate it to first order, and that's how you get the Stark effect.
Right.
Yeah, the linear Stark effect. You can go higher orders if you would like to calculate it with more accuracy, but the Games US program, from my understanding, uses the first order approximation.
What is E? Yeah.
What is E or Z?
E naught, like equation 1, the first term.
So E is just the energy of the system, and we're taking a Taylor expansion to
But
see how the,
which like DFT, Hartree-Fock.
you can use
Normally I'm using Hartree-Fock, you would use that. But I don't know.
Yeah. I use DFT, and like you're still adding a perturbation to the Hamiltonian. So I'm using a Consham Hamiltonian, right, for DFT, and then I just applied the perturbation on top of that.
Okay, so E naught is a DFT energy. Okay.
Yeah, this is E0 E naught for DFT.
Yeah. Okay. Yeah. Okay. Thank you.
No
Other
problem.
questions? I mean, just a comment, Jean-Michel. I mean, first order treatment under a high power is probably not accurate. I guess the thing is this already shows some strong perturbations. Is there any other questions?
I know linear, like the linear response TDDFT though, it's also a linear response thing. So
yeah.
It's very complicated.
Any other questions?
I have a question.
Okay, Kelly.
Hey, Mike. It's good to see you again, man.
Hey.
So I was nothing much. So thank you. I really enjoyed your talk, and I was just kind of wondering, like you were talking a lot about how when you do these structural types of pump probe experiments, like you have to pump the samples so hard that you know, you don't really know like where that energy transfer is going, because that was kind of like the gist of your whole presentation. More or less. And so I was wondering, like, you know, just like kind of comparing like the spectroscopic studies of like these types of systems or like any other systems, and trying to like draw correlations to like this structural type of pump probe. Like, generally speaking, like how much of a correlation is there, or is there still like a lot of work like this that needs to be done to be able to like, you know, draw definite conclusions?
I'm sorry, can you repeat it? It was really choppy on my end.
Yeah, sure. So basically I was wondering, like, you know, if I wanted to compare like a, you know, like a spectroscopic pump probe to like a structural pump probe experiment, and I didn't, and like you guys had to like pump these samples like really hard to be able to like get the signal that you saw, like how much of a I'm just kind of wondering, like, you know, how much work like your project like this has to be done to really like understand like how much of like that extra energy from the pump needs to be there to like be able to like, you know, draw conclusions to like what, you know, what state is really being like accessed and stuff like that.
All I know on that is you would need to do some type of dependence on the power density to figure that out. I'm not so well versed in experimental, to be quite honest with you.
No worries.
So I hope we can discuss it further in email if you'd like. My email's at the first slide.
Okay.
Maybe I just make a comment. So the motivation for these calculations was that people have used at XFELs, which are rather large experiments, extremely high excitation. Nobody would use the excitations that he used for optical studies. And what Jean-Michel shows is that they have multiphoton excitation. So the interpretation of their structural probe data is suspect. And when they talk about bleaching the sample and things like that, that's not what the end state. So we know there's some serious problems. They didn't access logic irrelevant. And so it's a real challenge for the XL community. They have to lower their excitation by a factor of over an order of magnitude more like 30, and it's not clear they have the signal to noise to be able to see structural changes under biologically relevant conditions. That's
okay.
They're 30 times to 100 times too high peak power, and they're hitting states that aren't biologically relevant. They have completely different photochemistry.
I see.
So this is a big deal.
Cool. Thank you. Yeah, thank
you. Any other?
Yeah, I have a question. You run Born-Oppenheimer molecular dynamics, but like as you said near the beginning, rhodopsin, the real system, has a conical intersection, so you wouldn't expect Born-Oppenheimer to be too great running dynamics in that region.
But we're not probing the dynamics at the conical intersection region. This is purely in the ground state near the far away from any conical intersection.
I see. Okay. I guess my general question is just even without considering the external magnetic field or electric field, could you benchmark just the standard dynamics against like more sophisticated like multi-configurational time-dependent R tree or something?
I did not consider those levels of theory. Like I didn't use anything like CASCF, especially CASCF for something like this would be way too overkill. It'd be computationally intractable in my opinion, or maybe I'm not well versed enough in CASCF to. I've done CASCF calculations, but even this is a 208 atom system, right?
Mm-hmm.
That's pretty large for a CASCF calculation, and using anything like multi-configurational would be heavy. It'd be really heavy, right?
Yeah, no, I was more so thinking just on your reduced model you used later on, but no, that makes sense.
Oh yeah, the reduced model might have been doable, maybe.
Yeah. Okay, thank you.
Any other questions?
I
think sorry, go ahead. Yeah.
I was just when you said you moved from D3 to I guess for your confirmation relaxation. When you remove D3, I know that what can happen is the conformation can change slightly because it no longer has that attraction or repulsion because of this dispersion added term. Did you repeat your relaxation without the D3 when you did your TD-DFT studies?
Yeah, so the TD-DFT part, I just extracted the coordinates that used the CPCM and D3, and then I took those coordinates and didn't do any re-optimization in those. It was optimized with the bigger structure already, which would be more accurate anyways because the bigger structure has more steric constraints and electrical interactions, right?
Okay, so just the reason I ask is you will have some change to just the potential your nuclei will feel because the repulsion terms will be slightly different from going with the D3 to no dispersion correction. That's just why I wondered if you would relaxed or anything like that.
Yeah, of course. If I could have, I would have, but I had to resort to not using it at all because it just wouldn't run. There'd be it would run the DFT calculation with was it D3 wasn't available for TD-DFT, but with CPCM the calculation would run in DFT, but then the TD-DFT would just show a bunch of like when the text doesn't know what it is, it's like all these boxes or question marks. Yeah, that would just show up and no numbers would show, and I was like, I don't understand what's going on here. I looked for advice on it. Nothing.
So did you? Well, I not too much of a. I've just done a few computations, so unfortunately probably couldn't give you advice. But I wonder if you redid the other calculations as well with no D3 correction, just so you would have a benchmark to say that you were justified in proceeding without this dispersion correction.
Well, the justification for not using the dispersion correction, I saw it in the what's it called? The optical absorption spectra that I produced, and it matched pretty closely to what I saw in experiment. So that was the justification that I was using for removing the D3.
Okay.
And also, as I said in the talk during, I showed you that the molecular orbitals involved were just of those three that were nearby the retinal, the tryptophan 82, tryptophan 182, sorry, 182, 85, and tyrosine 185, I think, or tryptophan 86, sorry, yeah.
Okay, thanks.
Yeah.
Okay, any other questions? right, so great. So thanks, Jean-Michel. I think it was a very nice talk, and you showed how pervasive high fields can be in manipulating molecular energies and states, and the care that's needed to do these things properly. So I think you've done a really great service to the community, and hopefully they'll listen. Thanks, everybody. Ciao.
Thank you for showing up.
Before we all get off, just a reminder from Mark to please fill out the feedback form on the Quercus. Thanks all.
All right, great. Thanks, everybody. Bye-bye.
Thank you.